Hello ladies and gentlemen, welcome to my YouTube channel. I'm aware that you are writing your combined science paper 3 on 29 November. We will need to prepare for that exam. Paper 3 is for practicals. I didn't upload in paper 3 before, but I will try my best to share the knowledge I have on practicals. I will give you some hints and tips on how to pass your practicals with flying colors. So today let us look at food tastes that are examined in combined science under biology section. So you're supposed to know how to taste food for starch, to taste food for reducing sugars, to taste for proteins, and then finally to taste for fats. You should know the procedures that are taken and also the observation that are going to be observed when those food are present. So first, let us look on how to taste food for starch. This is the procedure that we need to follow. A little starch is shaken in a test tube with some cold water and then boiled. We are going to observe a clear solution. After that, we need to add few drops of iodine solution. Iodine is brown in color, so if starch is present, it is going to change from brown to blue-black. If absent, it is going to remain brown as shown in the diagram. So this is what you should understand when tasting food for starch. Let us move on to the second objective. In the second objective, we want to look at how we taste food for reducing sugars. So reducing sugars, for example, glucose is an example of a reducing sugar. We need to add Benedict solution to about 5 cubic centimeters of food solution and heat gently in a water bath. So these are the colors that you are going to observe. If you observe a blue solution, it means that reducing sugar is absent. If you observe a green to yellow color, it shows that there are traces of reducing sugar. And then if you observe an orange color, it means that reducing sugars are present at a moderate amount. And then if you observe a brick red color, it shows that there is large amount of reducing sugar. This is shown in the diagrams above. So make sure that you understand everything about reducing sugar, the procedure, and also the colors that shows positive taste for reducing sugars. That is all about how to taste for reducing sugars. In our third objective, we want to look at how to taste for proteins. We use what is known as Beirut taste. It is used to detect proteins as it can detect peptide bonds between amino acids, which are structural units of proteins. So this is the procedure that we need to do. First, we need to add 5 cubic centimeters of sodium hydroxide on the food sample. After that, we need to add 5 cubic centimeters of 1% copper sulfate solution. If you observe a purple color, it indicates the presence of proteins. So this is what you should know when testing for proteins. The positive color for proteins is a purple color. Lastly, we will look at how to taste food for fats. The procedure is as follows. First, we need to place about 2 cubic centimeters of food solution in a test tube. After that, we need to add 2 cubic centimeters of ethanol and shake thoroughly until the fat dissolves. After that, we need to pour off ethanol into another test tube, and then we add 2 cubic centimeters of water to that test tube. If you observe a cloud white emulsion, it shows the presence of fats. So that is what you should know when tasting food for fats. The positive taste for fats is a cloud white emulsion that is going to be formed. So this marks the end of our tutorial today on food tastes that are examined in combined science. Let us meet again in the next session. I will be explaining the practical on food that was in paper 3 of combined science, June 2019. Thank you so much, guys, for following me on this channel. Don't forget to subscribe, like, and share my videos. I love you all. This is Eve signing out.